Good morning. We hope you all had a good night last night. Everybody found their way to whatever bed they were in and got back here, right? Okay, good. Good start. So a couple of sort of housekeeping items. One is that in the email we sent out this morning, there was one numerical typo. So no, Yoshibak will not be speaking during Barbara's presentation. It will be after. So just look at the flow. Today has got a lot going on, so allow us to kind of try and stay on schedule, but allow for a little bit of slop here and there. And in honor of Jamer's presentation last night, there he is, I am wearing this pink shirt, which I think was as close as I could get to the person in the park. And we have a gift for Jamer. As I said, I gave Helen a whole bunch of things that says this things go to 11 for our anniversary, and Helen wants to give this to you because she still doesn't want it. So there you go. You go. Yes, there you go. So, yeah, sometimes our personal lives bleed over into our professional lives, so there you go. So we have a busy day, so we're going to get going. We are thrilled to have Mike Levin here with us here. And he will be presenting with us. And yes, oh, whoops, sorry, I'm standing in front of the screen, but he can actually see you through that computer. So we do kind of have the max headroom as well as the screen thing. And Mike is going to give us a talk, and Elaine is going to join and show some slides, and we're going to have some nice time for Q&A and everything else. And other than that, I'm going to step out of the way and let you tell your story. I have to look at him this way. So go right ahead, Mike.
All right, let's see. Great, can everybody see my slide? Perfect. Okay, well, thank you so much for giving me the opportunity to share some ideas with you. I'm sorry I can't be there in person. Afterwards, if anybody is interested in some of the details, the data set, software, published papers, everything is at this site. And then my personal thoughts on what it all means tend to be here. So the main points that I'd like to transmit today are these. First of all, I'm going to talk about agential materials and in general, the field of diverse intelligence or problem solving in unconventional substrates. I'm going to try to widen a little bit our expectations of what active agents and what intelligence looks like. I'm going to use a specific example in regenerative medicine of cellular collectives functioning as a large-scale intelligence that navigates anatomical space. I'm going to be talking about bioelectricity as a kind of cognitive glue that binds these subunits together towards common purpose. And all of this is to show you an example of being able to recognize, communicate with, and actively engage with a novel type of intelligence that we're not really used to dealing with. It has many applications for regenerative medicine, which I'll show you, but in particular, it serves as an example of how what the benefits, the practical benefits are of some of the ideas that we're talking about. And then towards the end, I'm going to discuss some novel embodied minds. I'm going to show you a couple and talk about a few that are impending for all of us. And we'll talk a little bit about this notion of synthbiosis and the future of living positively with diverse sentient beings. And altogether, I think the whole talk boiled down into in one sentence is that we can use the collective intelligence of cell groups navigating the space of anatomical possibilities to show us how you can move certain philosophical ideas forward to therapeutics and then to some broader issues beyond. So this is a very well-known classic painting. This is Adam naming the animals in the Garden of Eden, and this is a kind of a conventional view with which most of us arrive at these questions. And there is something profoundly wrong here, but also something very deeply correct, and I want to talk about those. What I think is wrong here is that by looking at this, you get the idea that what we're faced with is a set of discrete natural kinds. So there are specific animals. We all know where they are, what the difference among them is. Here's Adam. He's able to count them. He's able to point to them. And by the way, he's significantly different from them. He's a different kind of creature. So those are the things I think are going to need to change, and I'll show you why. But the thing that's I think quite deep and correct about this is that in these old traditions, naming something meant that you've understood their true nature. And the idea of giving something a name or discovering its true name meant that you've understood something very fundamental about what it is and how you're going to relate to it. And this, I think, is profound because as individuals and as a society, we are going to have to be able to name in the sense a very wide range of highly diverse embodied minds. So the first thing I want to point out is that ever since we've understood evolution and development, we now know that what we're looking at is a continuum of beings. So each of us was a single cell, both on an evolutionary timescale and a developmental timescale, and that whatever philosophers might say about a human and the kinds of things that humans have, intelligence, goals, purpose, responsibilities, moral value, and so on, these are not things that snap into focus in some sort of sharp emergence, but are actually scaled up from properties of very different kinds of systems. And that developmental biology, neither developmental biology nor evolution offers any kind of a sharp phase transition at which, bam, you go from being one kind of system to something magical like this. So this sort of agential glow that surrounds the modern human actually spreads down into a spectrum. But now, not only that, but actually now with the advent of bioengineering and other kinds of synthetic sciences, we now see that there's another continuum as well. We stand at the epicenter of this other continuum where using changes to the biology, but also combinations of technology, we can produce, and in fact have and will increasingly produce a wide range of novel beings with different kinds of embodiments and different kinds of cognitive structures. And so again, anything that you want to say about a human, and you might be tempted to try to distinguish that from a machine, for example, you're going to have to be able to say something about all of these kinds of beings. You're going to have to say, well, exactly what is it that, and how much of these changes and what is it that makes them different and so on. And that's because biology is incredibly interoperable, and I'll say some things at the end about why, but basically at every level of biological organization, we can introduce engineered materials, engineered algorithms, and so on, making all kinds of chimeras and hybrids and so on. So what I'm interested in is developing a framework that will let us recognize, create, and ethically relate to truly diverse intelligences, regardless of what they're made of or how they got here. That means familiar creatures, so things that we think about, primates, maybe a whale or an octopus and things like that, but also all kinds of unusual creatures, colonial organisms, swarms, unicellular life forms, engineered life, so synthetic morphology, which I'll show you examples of, AI agents, either robotically embodied or pure software, and maybe someday even alien exobiological agents. I'm certainly not the first person to try for something like this. So here's 1943, Rosenbluth, Wiener, and Bigelow try to show a scale of how it is that you can get from purely passive matter through various cybernetic arrangements all the way up to this kind of human high-order metacognition and so on. So this is the sort of thing I'm looking for, and it's described in great detail in this paper called TAME, T-A-M-E, Technological Approach to Mind Everywhere. For me, what's really critical is that whatever frameworks we come up with, they have to be useful. They can't just be philosophy. They have to move experimental work forward. They have to lead to new discoveries and new capabilities, and as well to better ways to understand the ethics of increasingly complex life on this planet. So central to this kind of framework is this idea of an axis of persuadability. You'll note that it has some things on it on the same spectrum that you might have thought were quite different, which are machines, and then here are some humans and so on. And so the idea here is that all along the spectrum, what varies is the degree of autonomy and the kind of tools that you might bring to relate to that system. So here, for these kinds of machines, you're only going to be doing hardware rewiring. You cannot punish them. You cannot convince them of anything. And those are the tools that you bring. Here, the tools of cybernetics and control theory start to be useful because you don't need to rewire your whole heating system to get your house to stay at a different temperature. You just need to understand how the goal is encoded, and then you can set the set point, and the system will do what it needs to do. So more autonomy, and now a different way for you to relate to it. And then eventually you reach some systems where actually you really don't need to know much about the molecular mechanisms. Humans trained dogs and horses for thousands of years, knowing nothing about what was between their ears or what neuroscience was, because the system offers a completely different set of tools for interacting with it. So training, learning, and so on. And then, of course, up here you get into actually communication with the cogent arguments and things like this. So the important thing is that you can't just have feelings about where certain systems sit along this continuum. It's not one of these I'll know it when I see it kind of things. You have to do experiments because we're not very good actually at figuring out what kind of intelligent competencies something has until we study it and we test it. And so my claim here is that, first of all, anything you say about a new system, be it an AI, a language model, a robot, cells, tissues, if you're saying something about its level of intelligence and agency, you are, first of all, making an interaction protocol claim. You're saying, this is how I'm going to relate to that system using this set of approaches, and therefore you yourself are taking an IQ test because that might be a good idea or a bad idea depending on how things shake out. So, and this is experimental. We are not tied to ancient philosophical categories. We have to do experiments, and that's because while we are pretty good at recognizing intelligence in navigating three-dimensional space. So animals that are roughly medium-sized, they move at medium timescales and so on. We can sometimes see these things and recognize intelligence. But life explores, navigates, exploits, and suffers through all sorts of different other action spaces. There are the spaces of possible gene expression, physiological state space, and anatomical configuration space. All of these spaces are equally real and challenging to the embodied minds that navigate and traverse those spaces. So that means that all of this is not just about humans. It is about all kinds of other observers that have to understand how these systems navigate through those spaces. That means, parasites, conspecifics, the subunits of the system, greater systems within which they are embedded. All of these things are trying to understand where does a given system fit along this continuum. And we have to get beyond our evolutionary firmware that makes it very hard for us to think about intelligences in these other spaces that have much different scales and much different ways of navigating them. So the thing that we all have to start with is the idea that we were all an quiescent oocyte once. So you might look at that and you might say, well, this is a system that's perfectly describable by chemistry and physics. It's a little blob of chemicals, doesn't have any of the things that we normally associate with intelligence or agency and so on. But this process is slow and gradual, and that system will eventually become something like this, or even something like this, that's going to make statements about not being a physical machine and having extra properties and so on. And so this is the process that we need to understand. We need to understand the scaling of minds from very simple things that are amenable to chemistry and physics, all the way up through systems that really require all kinds of other new tools. And by the way, this is not the end of the story, as I'll show you, because there are ways that this simple subunit scales up to something enormous, but actually it can scale back down again and fall apart into pieces, or in fact transform into something completely different. So while all of us make this journey across the so-called Cartesian cut, we now have to understand the scaling. How is it that the cognitive light cone of these simple systems inflates to something like this? So the first thing to realize is that, yeah, we're all basically slowly amplified versions of much more simple biochemical systems, but at least we're a unified intelligence, right? We have this nice brain. We enjoy a pretty unified perspective on the world. We know that we are a self. We're not like a collective intelligence like ants and beehives and things like that, right? We're different. We're a truly unified intelligence. And in fact, Descartes liked this idea, and he liked the pineal gland inside the human brain because it was a singular organ, and he felt that the unified human experience needed a single structure in the brain to correspond to. But if he had had access to good microscopy, he would have found out that inside that pineal gland is all of this. There are tons of cells, right? And inside each of those cells is all this. So there really isn't one of anything. And that then reminds us that all intelligence is collective intelligence. We are all made of parts. This is a single cell, by the way. This is called a lacrimaria. And so you can see the kinds of building blocks that we are made of. They have their own agenda. They have their own competencies. And it's got a small cognitive light cone that basically extends a little bit or in time and space around this tiny region. And that is where its goals lie. It has physiological, metabolic, behavioral, and so on, and these goals. But we need to understand how this scales up into the goals of much more grandiose agents. And By the way, I don't have time to spend a lot on this, but just to say that even inside that cell, there are simple molecular networks that already can do things like Pavlovian conditioning. Intelligence does not, never mind the brain and the nervous system. We go back to cell level, but we even go back before that and point out that aspects of memory and learning are already here when you just have a small network of chemicals interacting with each other. That's already true, and we're trying to exploit that process here by developing applications in drug conditioning and things like this, where we can actually train cells to respond to drugs in particular ways, because they are already made of an agential material, not passive matter, not simple machines, but actually machines with learning capacity inside of every cell. And so now we see that evolution is using this amazing thing called a multi-scale competency architecture. Every level of organization in our bodies and beyond, to groups and collectives, has its own ability to solve problems in different spaces. They all have various competencies to navigate their spaces in an adaptive way. Intelligence does not merely show up at the level of brains and whole organisms. It is baked in all the way to the bottom through molecular networks. Now, Alan Turing was very interested in different types of embodiments of mind and machines that could think, and different other ways to do the kinds of things that humans do. So problem solving intelligence through reprogrammability. This is what he studied. But actually, interestingly enough, he wrote this paper, which was looking at how chemicals pull themselves together and generate order during embryonic development. Now, why would somebody who is interested in the intelligence of mathematics and reprogrammability and so on be looking at chemicals in development? And I think that's because he realized a very profound truth, which is that the mechanisms that are involved in the self-creation of minds are in fact very similar to the mechanisms involved in the self-creation of bodies. So we can learn from development, and we would have to, because that's where our minds arise, how it is that minds self-assemble. So I think he was onto a great truth. And one of the things that we can say actually is that if you look at an embryo, so this is, let's say, a piece of a flat chicken or mammalian bird or mammalian embryo. So you're looking down at this, and you'll say, okay, there's one embryo. It's an egg, and there's one embryo that comes out of it. Well, that's what normally comes out of it. But actually, if you ask the question, but how many selves are actually in there? The fact is that if we make little scratches in this blastoderm, and I used to do this as a grad student with duck embryos and use a little needle to put some scratches in it, what you end up with is little fragments that before they heal together, each one of them decides that it is the embryo and it self-organizes. And so you get twins and triplets and all kinds of things. So the real question to how many selves are inside of an embryo could be anywhere from 0 to probably half a dozen at least for the kinds of embryos that we know about. And they have to do this amazing self-construction. They have to know, where do I end and the outside world, or actually other beings, begin. And so this is very much not set by the genetics. This is revealing the self-construction that any living agent has to do to pull itself out of the kind of a background of disorder and establish its boundaries between its own self and the outside world. And this has many implications for issues of individuation, including split brain patients, dissociative identity disorders, and things like that. So what we're looking at here is the ability of an excitable medium in which the cells are going to act. What makes this one embryo is really that they all are they all share the same plan of what they are going to do, right? The path that they're going to take through anatomical space. They're all aligned and committed to the same story. That's what makes it one embryo. And these little islands here are committed to slightly different stories because each one of them is going to do it. So it's a matter of alignment to get to be one of these beings. And so let's ask, where does this information come from? Because we all start life like this, a collection of embryonic blastomeres. Here is a cross-section through a human torso. Where does this order come from? Look at this incredible pattern. I could just as easily have shown you brain architecture. I mean, it's an amazing pattern. Where does this come from? You might be tempted to say it's in the DNA, but of course we can read genomes now, and even long before that, we knew that the DNA doesn't directly set any of this any more than the DNA of termites set the structure of their colony here of this building, or the DNA of spider webs indicates the specific construction of the spider web. what the DNA encodes is the hardware. It encodes the little proteins, the tiny hardware that every cell gets to have. After that, it's the physiology or the real-time working out of this physiological software that allows the cells to figure out what to build and when to stop and so on. So we're interested in understanding how to rebuild. So if something is damaged, for example, how do we convince these cells to rebuild? And as engineers, we want to ask, but what else is possible? What else can these cells build besides their native kind of course that they take through anatomical space? So imagine in the future, this is kind of my vision of where this field is going. We want to get to something called an anatomical compiler. What that means is that someday you will sit down in front of a computer system and you will draw the plant, animal, organ, biobot, whatever that you want. It can be a standard one for transplantation, let's say, or it can be something completely new. It doesn't matter. You should be able to draw anything you like. And if we had this, what it would do is compile your target description into a set of stimuli that would have to be given to cells to get them to build exactly that. In this case, we've drawn a three-headed flatworm, and then here it is. But the idea, this is kind of the end game for the field. But we don't have anything remotely like this. Now, why do we need it? Well, first of all, because if we had it, all of this would go away. Right? This would be transformative regenerative medicine. So there would be no more birth defects. You could regenerate after traumatic injury. You could reprogram cancer, defeat aging and degenerative disease, if we had a way to communicate to cells what it is that they should build. Okay, all of these things would be solved. And so that underscores an important aspect of this, that this anatomical compiler, it's not a 3D printer. It's not anything that tries to micromanage cell constructions onto a scaffold or anything like that. It's a communications device. It's a translator. It tries to get your desired spec as the bioengineer. It tries to get your spec into the recorded set points that the cells are using to decide what they're going to do. And so this is why we don't have this yet, despite the fact that genomics and biochemistry and molecular biology have been going back gangbusters for decades. It's because we have not yet understood how to communicate to cells. So while the state of the art is very good at this kind of stuff, different kinds of genes and proteins interacting with each other, we're still very far away from this. You know, if you want to regenerate a limb or fix a birth defect or have a different structure, and that's because biomedicine has been largely focused at the hardware. Okay, so genomic editing, protein engineering, pathway rewiring, all those exciting things look like this. This is where computer science was in the 40s and 50s. If you're going to rewire it, you would have to physically get in there, and if you wanted it to do something different, you would have to physically rewire it. And that is the assumption that what we're dealing with is merely a complex machine. It's complex, but it's a machine. That's the standard assumption of molecular biology and a lot of biomedicine. But I think this is wrong, and I think we're fundamentally dealing with an agential material. And once we really realize this and deploy the tools that are used in behavior science and cognitive neuroscience, we will then be able to do for medicine what computer science has become in information technology, where on your laptop, when you want to go from Microsoft Word to Photoshop, you don't get out your soldering iron and start rewiring, because we now understand that if your hardware is good enough, you can do way better than this. You have high-level ways of communicating with your device, and the biological hardware is absolutely good enough. And so I want to show you some examples. You know, we started out talking about intelligence and navigating the anatomical space. So what are the capabilities of the biological hardware? Well, first of all, here's development. So here's a human egg, gives rise to a human body. This is an amazing rise in complexity, and it's very reliable, but that is not why I call it intelligence. This is really important. It's not just because complexity rises. It's very easy to increase complexity with no intelligence. That's not it. It's because this process has some amazing problem-solving competencies. One of those is that if you cut this early embryo in half, you don't get two half bodies. You get two perfectly normal monozygotic twins. Because the whole thing, and as I showed you for those duck embryos a few minutes ago, the whole thing is a decision-making process of finding borders and navigating towards your anatomical goal state. It is not a mechanical kind of a hardwired step through a bunch of biochemical steps. That's not what's going on here. And this system, once you've cut it in half, each side realizes that a bunch of it is missing, and so they can all get to. So you know, if you boil down anatomical space into just two axes, this is the goal space corresponding to the goal states corresponding to the kind of a normal human target morphology. They can all get there from different starting positions, just avoiding certain local maxima and so on. That is the navigation I'm talking about. Now, that process in some animals continues throughout their lifespan. So this is a salamander, a kind of an amphibian called an axolotl. They regenerate their limbs, their eyes, their jaws, their portions of the heart and brain, the spinal cord, and so on. And you can see that when they bite each other's legs off, no matter where it is amputated, they will continue to, the cells will very rapidly grow and eventually give you a perfect axolotl limb. Now, the most amazing thing about this regenerative capacity is that it stops. When does it stop? Well, it stops when a correct salamander arm has been completed. So what you're looking at here is a kind of anatomical homeostasis, like the thermostat in your house, which continuously seeks to reduce error, so relative to a set point. So it has a memory, it has to remember what the correct point is. But here, this is what it's remembering is this particular structure. It's going to, the cells can keep going until that structure is rebuilt. So it's the ability to get to the same point from different starting positions, even when you're deviated from it. And you can think of all of development as just regeneration. You know, development is regenerating an entire body from just one cell, right? So all of this is about error minimization and about navigating that space to get to where you know you need to be. In fact, now this is one of my favorite examples. This is just remarkable. If you can take a newt and make sure that during the earliest steps of cleavage, the DNA multiplies extra, so the cells become bigger, right, to accommodate those extra copies of the genome. And when the cells get bigger, the newt stays the same size. And so taking a cross section through the tubule, you then see that instead of like 8 to 10 cells, there's now fewer of them that still make exactly the same structure. And when you make when you make newts that have so many copies of their genome that the cells become enormous like this, then one cell will wrap around itself and leave a hole in the middle. So what you're seeing here is this incredible problem-solving capacity where if you're a newt coming into the world, you can't count on how many copies of your genome you're going to have. You don't know what the size or the number of your cells are going to be. You have to figure out how to solve your problem and get to where you're going using the tools that you have. Now, this is one set of tools, cell-to-cell communication. This is cytoskeletal bending. It's using a completely different set of molecular steps to achieve your goal. This is but you know, textbook intelligence is using the tools you have to solve the problem under completely novel circumstances. So not being able to assume and not be able to take your evolutionary past literally is really critical for problem solving. And so now we see that what happens in the creation of bodies is this incredible problem solving capacity where you can do all sorts of things and they will readjust and get their goal met. So how is that possible? What is doing all these computations? And in particular, where is the memory stored? You know, how do these things know what shape they're supposed to be? So a couple of decades ago now, we started in my group developing some of the first tools that were used to discover this. And the idea is we took our inspiration from the nervous system. In the brain and in the nervous system, which is a kind of the one non-controversial example of systems that have goals and have clever behaviors to pursue those goals. This is how it works. The hardware is relying on ion channels to set voltage states in these neurons. And these electrical voltage states do or don't get propagated to their neighbors through these electrical synapses. And so as a result, you get this very dynamic electrophysiological activity. Here's a zebrafish brain recorded by this group showing you what happens when the fish is thinking about whatever it is that fish think about. And you could imagine, as people are now doing, this kind of project of neural decoding. Could we read and decode these physiological events to understand what are the memories, the preferences, the goals, the behavioral repertoire of this animal? Like all that cognitive content is here in this electrophysiology. That's what neuroscience is committed to. So it turns out that this system, this kind of bioelectric system for processing information towards goal-directed activity is ancient. It is not something that was new with brains. Every cell in your body has these ion channels. Most of them have these gap junctions that allow the networks to form. And we can actually then use these tools to do a kind of a non-neural decoding and ask, what are all these cells talking to each other about? And what does the collective remember? And what does it know that the individual cells don't know? So we developed some of the first tools to molecularly read and write this electrical information. So here it is using voltage-sensitive fluorescent dyes on a bunch of cells, and you can see all the amazing electrical conversations that these cells are having. Some of them are fast, some of them are slow, but this pattern is just like in the brain. This pattern encodes the collective processing of information in this group of cells. And then, of course, we do a lot of computational modeling to try to understand how that works. And let me show you some specific dynamic examples. So here is what we call the electric face. This is again a voltage sensitive dye showing you what happens when a frog embryo is trying to put its face together. So this is one frame from that video. And what you see here, I mean, the thing looks like a face. This is why we call it an electric face. It's very easy to see that these bioelectric states, in fact, map to here's where the eye is going to go, here's where the mouth is going to go, here are the placodes out to the side. So what we're seeing is the pre-pattern. We're seeing the informational scaffold that these cells are going to use to turn on specific genes and ultimately create a vertebrate face. There are also, so that's and as I'll show you in a minute, this pattern is essential to being able to reach that goal. Now, there are also patterns that are pathological. If we introduce a human oncogene into these tadpoles, the oncogene will make a tumor, but the way it does it is by disconnecting cells from the electrical network. So here you can see this is where the tumor is going to be, and there's a bunch of escapees, straggler cells here that are going to basically disconnect from the network, roll back to their unicellular lifestyle, and just treat the rest of the body as external environment, right? That again, that border between self and world, evolution scales it up, but during this cancer process, it can actually scale back down to the tiny cognitive light cones of individual cells, which then do not recognize that they're part of this large network, and that has many implications for therapeutics, which we are pursuing. So I've shown you how we detect these patterns, but of course, more important than detecting is actually being able to write them so that we can show what happens. And so we don't use, there are no applied fields, there are no electrodes, there are no waves, frequencies, electromagnetics, nothing like that. What we do is we manipulate the native interface that these cells are normally using to hack each other. So those are the ion channels in the membrane and these gap junctions that allow the cells to talk to each other. And we use all the tools of neuroscience, which is pharmacology and optogenetics and all these other tools to control the electric state of these cells. And so now the million-dollar question. Having posited that this electrical connectivity is the glue, much like in the brain, it is the glue that binds individual cells together into these grandiose pattern memories of making limbs and eyes and brains and so on. What actually happens when we perturb these pattern memories? Can we test this idea, right? How do we know this is right? So I only have time to show you a couple of examples, but here's one. I showed you that little spot of voltage depolarization that indicates where the eye is going to go. And so if you actually inject some potassium channel RNA into other cells that are going, this is in the frog model, this is a tadpole here, that are going to make other structures, you can set up an ectopic little domain of voltage that says build an eye here. And when you do that, sure enough, that's what the cells do. So there's an eye sitting in some gut tissue. If you section them, this eye has all the lens, retina, all that stuff that eyes are supposed to have. And all of this was done not by micromanaging. We did not control the stem cells. We don't have any idea how to build an actual eye from scratch. We didn't need to do that. We communicated to those cells, you should build an eye. This is an example of using that bioelectrical interface to communicate with with a different kind of collective intelligence. It doesn't navigate three-dimensional space the way that motile animals do. It navigates anatomical space, and we were able to pass it one message: make an eye right here. That's it. It did the rest. It was competent to do all the downstream steps. By the way, if you only get a few cells here, what they will do is recruit all their neighbors. The blue ones are the ones we injected. All this other stuff, we never touched it. These cells induce their neighbors to help them complete this construction project, much like I'm sure you've seen ants communicate and recruit nestmates to help them move a large piece of food or something like that. All of this is part of the competency of the tissue. We did not have to build that in. Another example of communication occurs in our regenerative medicine program. So frogs, unlike salamanders, do not regenerate their legs. So what can we say to these cells to get them to rebuild a leg, even if we don't know how to do it. Well, we discovered a bioelectric cocktail where 1 day, so 24 hours of treatment, gives a year and a half of leg growth, during which time we don't touch it at all. And so here you can see what happened immediately. Within 45 days, there's already some toes, there's a toenail. Here's some pro-regenerative genes are coming on, and you can see the leg is touch sensitive and it's motile. The animal can feel the touch and can use that leg to swim. So this is, again, the idea of not micromanagement, but actually communicating that you should go not the scouring route, but the leg building route, and letting the system take care of the rest. At this point, I have to do a disclosure. So Dave Kaplan and I have started a company called Morphoceuticals Inc., which is trying to move this towards clinical applications, first in rodents and then hopefully in human patients to regenerate organs. So I show you this. The details aren't super important, but what is important is the idea that when we take seriously that the dynamics that we're seeing are not just a mechanical clockwork, but are actually a set of decision-making processes by a competent system that is trying to minimize error relative to set points, it unlocks all kinds of discoveries that are inaccessible if we treat the thing as a dumb machine. So, and this is, I'm sure this is just scratching the surface. So the next thing I wanted to show you was, when we're looking at these examples of repair, and there are many examples of birth defects and many other kinds of things that we've done, it's very clear where the pattern comes from. Presumably, it comes from evolution. In other words, that is the correct shape for that species, and so that's where the pattern comes from, or so the conventional story goes. But we wanted to push that idea, and the way we did it was to try to make a synthetic novel life form that is in a different configuration than anything that in its evolutionary lineage. And so the way we do that is we take early frog embryos, we take some of the epithelial cells from the top here, we put them by themselves, we dissociate them, and we put them by themselves in this little depression. Now, many things could have happened. They could have died, they could have crawled away from each other, they could have made a flat monolayer like cell culture. But we wanted to know, what would these cells do if we extract them from the embryo and give them an opportunity to reboot their multicellularity? Well, what they do, it turns out, they don't do any of these things. They gather together and they make this thing we call the xenobot. Xenobot because Xenopus laevis is the name of the frog that we get them from, and we think this is, among other things, a biorobotics platform. So they start to swim. They're autonomous. They use little hairs on their outer surface. These hairs are normally used to move mucus down the body of a frog, but here they're using it to row against the environment. They can go in circles. They can patrol back and forth like this. They can have collective behaviors where they interact with other bots. Here's one moving down this maze-like structure. You'll see here that it takes this cordon without having to bump into the opposite wall. And then here it spontaneously turns around and goes back where it came from. Remember, there's no neurons here. This is just skin. This is a novel proto-organism that's made entirely of skin cells here. It has all kinds of interesting behaviors. If we look at its electrophysiological activity, so this is a calcium sensor, there's tons of it. This looks actually like the kind of thing that people record from brains, even though there's no nervous system here. And then if we were to actually functionally analyze these signals, there are some really interesting features of them that this is just a preprint for now, but there are some interesting comparisons that can be made between the physiology that goes on here and the kinds of things we would read from a brain. They have some unexpected capabilities. Here's something we call kinematic replication. We've made it impossible for these bots to reproduce in the normal froggy fashion. They don't have any of the organs that are needed to do that. But what they can do is if you sprinkle a bunch of loose skin cells into their field, so this white stuff is just loose epithelial cells, what they'll do is they'll run around and they will collect them into little piles, and they'll polish the little piles, both collectively and individually. And then because they're working with not passive matter, but an agential material, these little piles mature into the next generation of xenobots. And guess what they do? They run around and make the next generation, and they make the next generation, and so on. What has the frog genome learned here? What do genomes do? Well, over the years of evolutionary selection, it certainly learned to do this. So it can make a specific developmental sequence, and then some tadpoles and so on. But it also makes xenobots with their own really weird developmental sequence and things like kinematic self-replication. As far as we know, no other animal on Earth reproduces via kinematic self-replication. There's never been any selection to be a good xenobot. Um, there's never been any xenobots, and so this immediately raises the question: the goals that these collective systems have, and now this is now working up towards the kind of the broader thing we're going to talk about, AIs and so on. The goals of these systems are not obvious, and the goals of novel systems, which do not have a history of evolutionary selection, are even harder to predict. And so, what's really necessary now is a science of understanding what are the goals that novel collective systems are going to have. And by the way, their behavior, I haven't said anything about their behavioral intelligence, but over the next few months, we'll have a couple papers out that actually characterizes that. I'll show you another quick example, which is this. If I just showed you this video, you might guess that this is some sort of primitive organism I got from the bottom of a pond somewhere. We have no idea what its goals or its competencies are. Turns out this is something we call an anthrobot. It's made out of human cells. So if you were to sequence this, this is 100% human, normal human genome. This comes from adult patients. These are tracheal epithelial cells that come together to be an anthrobot instead of a xenobot from the frog. And so this is a total reboot of human multicellularity. Again, it doesn't look anything like this the standard organism, but they have some amazing properties. If you put them on a petri dish where you've grown a bunch of human neurons and you put a big scratch through it, right? So here's the defect, a neural wound here. What you'll see is that these bots cluster. They form something we call a superbot. And underneath, within 4 days, you lift it up. What they've been doing is taking the two edges of the neural wound and healing it together. Okay? Now, who would have known that the cells that sit quietly in your trachea for long periods of time, when given the opportunity, will rebuild themselves into a self-motile little creature that has the ability to go around and heal neural wounds? Who knows what else it can do, right? So, okay. So to kind of start winding down here, I want to point this out. Because of this ability of life forms to decide on the fly how to solve various problems. They're not hardwired, at least most of them are not. They are constantly using fairly ingenious problem-solving competencies to get their needs met. It means that not only can they regenerate after damage, but life is incredibly interoperable. Almost any combination of evolved material, engineered material, and software is some kind of viable agent. So we have cyborgs and hybrids and chimeras. Many of these things already exist. They will increasingly be more prevalent. All of the variety of biology that Darwin saw when he said that, when he used this phrase, endless forms most beautiful, yeah, that's one tiny corner of the space of possible beings. And we are going to be living with all of these. So this will increasingly be a part of society, not because, and the point of all this is not just, to make weird creatures and just to sort of see what we can do in an engineering sense. The changes that certain people will make to themselves and to other systems, both biological and technological, are shedding critical light on understanding what are we. What does it mean to truly have a mind, to have goals, to have intelligence, to understand when we say, well, this thing doesn't really understand, well, I really understand. Like, what does all that mean, for a collective of cells to say that they understand something and they can navigate a specific space? So all of this is about understanding us better, but critically, it's about being able to ethically relate to a wide range of beings that we have never seen before and are not on the evolutionary tree with us. And so just two very, very quick things I will say is that a lot of people have strong feelings about what certain types of AIs can and can't do and what living things can do and so on. And I want to point out that it is really essential at this early stage of the game to have some humility about this, because we do not know what it is that allows biological machines, so constructs of biochemistry and so on, to enjoy the kinds of cognitive properties that we all have. We actually don't know. And therefore, we have to be really careful about saying what other kinds of constructs can and can't do. In fact, there's been all kinds of research in minimal matter and our own work on very simple sorting algorithms. If any of you are coders out there, it's something you study in your first year of algorithms in the computer science curriculum, of these sorting algorithms. We found novel problem solving capacities in simple sorting algorithms that nobody had noticed for a really long time. And so it really doesn't take much to start to climb that continuum of agency. And we have to, I will argue that for the same reason that you can't look at the laws of chemistry and think that you've understood everything that's important about being a cognitive being in the world, you really can't look at the construction of a machine, either in its hardware or its algorithm, and think that you've understood everything that it's doing. Unexpected, not just complexity, not just unpredictability, but unexpected cognition starts to crop up extremely early, and it doesn't even require cells, never mind brains or anything like that. And the very last thing I want to say, I'm a couple minutes over, but the very last thing I want to say is this: I want to tell you a quick story. This is a science fiction kind of a science fiction story, but I think it makes an important point. Imagine that some creatures come out of the center of the Earth. They live in the core of the Earth. They are incredibly dense. They come up onto the surface. What do they see? Well, they don't see any of our stuff. They don't see any of this. I mean, they're using gamma rays as vision because they have to see through in rock. They don't see any of this. What they say is that, well, this planet seems to be covered by like a very tenuous, very ethereal plasma. There's some kind of gas that's on top of them, but none of this is solid to them. They are solid. All of this is basically gas. They're sort of walking all over everything. And one of them is a scientist, and he's been watching this gas, and he says to the others, I've been watching this gas. There's patterns in this gas. And you would almost swear that these patterns are actually doing something. You almost think that they're agents of some kind. They seem to have goals and they solve problems. And the others are saying, well, that's ridiculous. Patterns in the gas can't be agential. We're real. They're just patterns in an excitable medium. And by the way, how long do these patterns hold together? He says, that'd be about around 80 years on average. And well, that's nuts. Nothing interesting can happen in 80 years. And so this kind of thing reminds you that, and this has been understood by people working in metabolics and so on. I mean, we too are temporary patterns in metabolic media, like whirlpools and solitons and gliders in the game of life and so on. All agents are actually patterns in some medium. And so we have to be really, I think, really careful in our commitment to expanding our ability to recognize novel minds, whether they be in physical embodiments like ours or something really very alien. So I'll end here, and just to point out that I think intelligence is probably very widely distributed throughout the world. Learning to rise above our evolutionary limitations and recognize it, I think, is critical to expand this idea of sentient beings. I don't think we've even scratched the surface of what that really is. We now have the field of diverse intelligence has a research agenda, principled search for frameworks that avoid teleophobia, but also avoid unwarranted animism, painting up the hopes and dreams on every rock. We don't have to daydream high or skew low. We can get it right, or at least we can optimize way past what we have now by dropping untenable ancient categories that are not serving us any longer. These things that are trying to that sound like we know what they are, machines and organisms and so on. It sounds like we know what we're talking about. We really don't. And so this is the beginnings of a research program that can use ideas of a continuum of agency, observer relative models, and I think AI as universal translators to these diverse intelligences to really have a much better future than is possible using today's frameworks. So if anybody's interested, I can send any number of papers where we go in depth on all this stuff. These are the people that contributed to all this work. So lots of postdocs and grad students and undergraduates and tech support. We have many amazing collaborators here. Our funders that I thank for supporting this work. And again, disclosures, there's 3 companies that are supporting us and are commercializing some of our IP. So yeah, I'll stop here and thank you so much for listening.
A fast start to the day, correct?
Thank you for 2 really fascinating talks, stimulating. So one of the takeaways I have from especially Michael's presentation is we're used to distinguishing software and hardware. You've shown that experience alters both. And I wonder if those are distinctions that we need to keep. It feels to me like it's in the interactions of experience and the whatever structure there is that comes with biology and whatever, I'm not sure what the software is, whatever is running through. But that distinction, especially the software hardware, because one can change the other, and experience changes both. So I think we need new ways of thinking about those things. So I'll let you answer that.
Yeah, thank you for that. Great point. So I think what's useful about the software-hardware distinction is simply the reminder that some hardware is reprogrammable. So this is something critical that computer science has given us, is this idea that for a wide class of machines, the things that they do and the things that they're capable of, the story is not told by an account of their physical medium. Okay, that they are reprogrammable and so on. So that's the useful part of it. But there's a framework that Josh Bongard and I are working on called polycomputing, which is all about how things appear to various observers or various other agents that interact with something. And the thing about software and hardware is that there is a standard way of looking at it. So let's say a simple Turing machine. You know, you've got this machine that writes data on the tape as it performs its computations. The conventional way is to say, okay, the machine is the actor here, and the tape is just it's a passive. The data is passive. It's a record of what's going on, of the computations that are done. But if you want to look at this from the perspective of the tape, you say, no, it's the patterns on the tape that are the agents. The machine just does whatever the tape says, right? And so this is something that we're wrestling with in our work on bioelectrics, which is that you can look at the idea, you can look at it as, okay, the cells are the agent and the bioelectrical patterns are the thoughts that this agent has about where in the anatomical space it's going to go. So the bioelectric patterns are the memory medium, and they are the cognitive content of the agent, which is the cells. Or you can turn it around and say, no, actually, it's the bioelectrical patterns themselves, and this is why I put that bit in the end about patterns being agents. And we could talk for a long time about this, but the idea that perhaps it's the physiological patterns that are the agent here, and the rest of the body, so the cells, the molecular networks, the chromatin modifications, all the gene expression, all that stuff, that's the tape. That's a scratch pad of the bioelectric patterns doing their decision making in the physical world, right? So the perspective matters, and each one of those views suggests different research programs, different approaches to therapeutics, different ways to think about what we are and where and how our perspectives affect what we do. But yeah, I don't think we need to throw away the hardware-software distinction. I think we need to keep the programmability aspect of it. And I think that really critical, we need to realize, as per this polycomputing framework, that there isn't one objective, correct story about what anything is or what it can do, but there are multiple useful stories told by different observers who will interpret physical events in different ways. And that allows us to squeeze even more use out of this question of, okay, so what's the computing device and what are the patterns that both move through it and direct everything that happens after that.
Lucas, go ahead.
Thanks for the great presentation. Another question for Michael. If the anatomical compiler is the end goal, what would be the 2 or 3 milestones that you would be extremely excited about? Something that shows we made substantial progress? Maybe just like the synthetic cell by Venter or something like that. And then maybe just as a nerdy add-on question, the modeling language of the compiler, do you think it will be imperative or declarative?
Sorry, I missed that last part. What about the language?
Of the anatomical compiler, will the language be declarative or imperative? Meaning, my question is, do you think we can operate with the systems only if we understand all the parts and how they create it? Or can we be declarative, saying, I want this kind of behavior from this system at this level, and I don't care about how it's implemented in a way? It's more of a question whether you think how deep we have to understand the system in order to program and work with it. But it's a little bit nerdy. So maybe just the milestone question is the one that I would be interested in.
Yeah. Well, so the kinds of so for example, the kind of things that Elena just showed you are a critical component of this, because learning to train the cells, in her case, the neural cells, and in other case of other people in the lab of non-neural cells, establishing that, I mean, training something is an excellent form of communication. You know you've communicated when you've trained something, and so that is an important early milestone for showing that we are able to motivate the cells to adopt novel behaviors and the cell collectives, and that then gets to your second point, which is that the language that we're going to use, we actually are trying to develop a form of a computing platform called goal-driven programming, where the idea is you are absolutely not declaring the steps that you wanted to go through. You have no idea what the steps are to go through. In some cases, the system is much better than you are at figuring out what to do. What you are trying to lay down is a set of incentive structures to communicate your goal, and to, in the case of very simple systems, to basically just train it with rewards and punishments. And in the case of much more advanced systems, to try to get aligned on goals and be part of a cooperative, collaborative system. I mean, I see a lot of our biomedical approaches as ultimately being a collaboration with the tissue and a set of communication events, not trying to force it or micromanage it or anything like that. So, so I think the kind of thing that Elena just showed you is a really important first part, to be able to communicate specific goals to tissues. But also important would be an understanding of the various competencies. We've just scratched the surface. You know, what I've shown you before about cells figuring out, hey, there's not enough of us to build this, let's recruit some friends. That whole conversation that they're having, because the other cells are saying, don't make an eye, we're skin, we're gut, you shouldn't make an eye. They're having this discussion. And so the ability to eavesdrop on those conversations, to translate them using machine learning tools and some other approaches, and to be able to make predictions and say, I see where this conversation is going, it's going to decide to do this or that, and then here are some stimuli that we can give it to try to coax it to go down this other route. Like all of those things, and we have a whole breakdown in addition to the cell interaction platform, which Elena just showed you, but also the kind of the computational models that it takes to model cells not as a bunch of molecular pathways that kind of mechanically roll downhill, but actually systems that do error minimization, that active inference, all the stuff that we know from neuroscience that happens all throughout your body. Those are some important next steps. And then there's all kinds of specific deliverable milestones along the way. You know, can we regenerate this or that? Can we normalize tumors, which we've done in some animal models? Can we repair certain kinds of defects? And you know, then that's how it goes forward.
Cool. Thanks for the really thought-provoking presentations, Mike and Elena. My name is Mike as well. I was just kind of curious, you know, how far would you be willing to push this kind of continuum of scale? You know, like is there any reason not to think about intelligence at like the planetary or solar system level, galaxy, universe, or, you know, conversely, like down to the molecular or atomic level? And is there a good framework for like thinking about what scale is appropriate to think about this stuff on? Like why talk about it at the cellular level and not, you know, molecular or organ level?
Yeah, great point. So first of all of these scales are potentially simultaneously cognitive. So we have done things at the molecular level. I absolutely do think that certain kinds of molecular events not only do learning and memory, but they have different simple forms of agency as well. So yes, we should be thinking about all of that. Now one critical thing that I will claim is that what we can't do is what some people do, which is they say the whole universe is a giant mind. I mean, maybe, and I'm actually very sympathetic to that view. But what's critical is you can't just assume it. So what you have to do is you have to do experiments, which means, and this is why, I mean, I showed you one model system, which is cells, because that's much easier to do these kind of experiments. I'd like to try training the weather and the, many other things, but those are like not really, tractable at this point. But for all of these things, you have to do experiments, which means you have to hypothesize what problem space it's operating in, what are its goals, and what competencies it has to fulfill those goals. And then you do perturbative experiments. So like in our case, you put a barrier between it and its goal. You change out some parts or something in the environment. And William James had this great definition of intelligence, which is same goal by different means, which is an intelligent system has some degree of competency to get its goal accomplished, even when stuff changes, right? And so now the question is, okay, how much competency? So making those hypotheses and then testing them is how you find out that you're dealing with a kind of intelligence, and more importantly, what kind. Now, that raises a couple of interesting points. First of all, how do you know when you yourself are part of a larger intelligence, and can you ever know? So I had somebody draw a cartoon for me once for these talks where there are two neurons in a brain, and one of them's kind of a materialist and the other one's kind of a mystic, and the first one says, boy, we live in this cold, unfeeling universe, and it's just mechanics, and there's no sense in it. There's no mind. We're just chemical machines. And the other one says, I don't know. I got this feeling. I can't prove it, but I've got this feeling that there's a greater order to things. It's almost like the universe wants something, and sometimes there are these waves back propagating through us, and they kind of hurt, and there's got to be a reason for it. And I kind of feel like it's learning something. And the first one says, you're crazy. And so now this raises an interesting question, right? Like going down is easier. I mean, it's hard. It's really hard. We've been, the people in this field have been fighting for a really long time to make these ideas take hold. But still, it's easier because because when you are trying to when you are a higher level intelligence looking at your parts, you have some hope of doing experiments. Going upwards is hard, and my gut feeling is, I can't prove this, but my gut feeling is that there's going to be some kind of Gödel-like theorem that is going to say you really can't ever know definitively that you're part of a larger system. If you're a cell, can you know definitively that you're part of a brain and what that brain wants? I don't think you'll be able to know definitively, but I do think that we can develop tools to gain evidence for it. And so now one might ask the question, well, what does that look like? When you're a part of a larger system, when things look mechanical to you, but there is some sort of global order that you're not able to catch on to because you're just a tiny little part of this like enormous thing, what would that look like? And I really don't think we have a good vocabulary for it, except that the closest that I think anybody got was Pauli and Jung and their work on synchronicity. I think that it would look exactly like synchronicity. It would look like events that are meaningful in some global context, but that you can find no direct causal relationship for at your level of description. You would find these large-scale patterns. I don't think you can prove anything, but I think you could get evidence for some sort of useful global pattern that is, basically what happens in these layers is that each layer is deforming the option space for its parts. And so the parts are just rolling down the hill. They're just minimizing energy and doing these kinds of things. But that's because the whole landscape was now deformed by the system above it. And it's often very hard to see that when you're in the landscape. So that's as close as I can get to this very profound question. Is I think, is it possible that actually with some friends, I once tried to design a solar system scale gravitational synapse. Like you could imagine setting up a gravitational system where, every time something comes flying in, that's your impulse or that's your bit, and then things change. And the next time it comes in, there's some hysteresis and now it's learned. And you can try to set up these things. So is there any reason why not? Absolutely no reason. I think we should be completely open to radically different embodiments, different scales, different materials. In fact, no materials at all in some cases. But going beyond just the possibility that that's true and gaining evidence that that's happening is going to be really hard for these larger systems. Not impossible, but I think really hard.
Josh.
Super fascinating. Thank you both so much. This is a question for both of you. I love on the intelligence continuum that there are no hard divide lines. Brilliant. I'm curious though, in either of your work, if you find there to be certain thresholds or tipping points, and I'll describe this in 3 different ways, but it's all the same thing, between reaction and prediction, or feed forward into backpropagation, or say homeostasis into allostasis. And I'm curious, again, where that jumps from sort of like the cell or the xenobot or the anthrobot that collects and recruits around it in this very forward-facing, immediate reactive, as you said, not intentful, into something like what we do as a human being, where we are forced to live in the past. We're always lagging, microseconds behind the reality. We are only able to respond to the near recent and the aggregate buffer. So I'm curious, again, in that continuum where there are no hard divide lines of intelligences, do you find that there are tipping points or thresholds where it sort of transitions into more predictive behaviors?
Elena, do you want to talk first? Okay. 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 Well, the first thing I want to say is that basically every time we've looked for, so you can absolutely make a scale with kind of different grades, and as Wiener and Rosenbluth did and Bigelow, of different scales of cognitive competencies. And what I know is that every time we look, we are A, surprised to find them, and B, we find more than anybody thought that was in there. So I think we have to be really careful. I mean, the one thing I won't claim is we haven't found anything that looks like language in these other models, but again, that's probably because we're not smart enough to look the right way. But below that, for example, active inference, perceptual control, visual illusions, memories, counterfactual memories, representation, all of these things, you can find them very low down on that scale. If you're willing to look at other spaces and to change scales, you can find these things all over the place. So even yeast and bacteria have predictive capacity. They have we just put out a preprint where we show that the Volvox, which is an algae, and so this is the work of grad student Franz Kuschling in my group. He found that algae can be surprised, and they don't like it. And in order to be surprised, you have to have expectations, and then you have to have the wherewithal to be ticked off when those expectations aren't met. And algae are already there. And I think actually it's way below that. So yes, I think there are kind of these we have categories that we use, but I think they show up very early in the complexity tree of things. I mean, there's a few interesting ones that I like. For example, if we think about I use this concept called the cognitive light cone. So the cognitive light cone for any given system is the size of the largest goal that it can pursue. Not how far it can sense or how far it can act, but the largest size of goals that it can pursue. And one interesting thing about the cognitive light cone is that possibly uniquely, we don't know, but possibly uniquely, humans have a cognitive light cone that's larger than their own lifespan. So if you're a goldfish and your goals are X minutes into the future, you're very likely to survive for that time. So your goals are actually achievable, right? For many humans, and if you have a dog, the cognitive light cone, certainly it has some memory, some predictive power, and maybe it cares about what happens in the yard of a certain size. You're never going to get your dog to care about what happens 3 months from now in the next town over. It's just, they can't do it. So I think there's a particular, there's something interesting that happens when your cognitive light cone is actually larger than your own lifespan, where you realize that, wait a minute, some of my goals are certainly not achievable in my own lifetime. What does that mean for me? And what are the stressors, psychological stressors that gives rise to, but also cooperative and long-term thinking and things like that. So there are other steps. For example, in the Buddhist tradition, there's this thing called the bodhisattva vow, which is a specific. It's a kind of a meta thing. It's a specific commitment to enlarge your own cognitive light cone. So the idea is that right now, given how we are right now, we have the ability to care about a certain number of others practically care about a certain number of others, and somebody, if something's happening to, 10 million other creatures, you're not 10 million times more sad than you are when it happens to one. I mean, we can't scale our compassion in that way. But there's this idea that you get to a certain point, and now you can actively work on enlarging your cognitive icon, whether that means it, like in the old traditions, through psychological practice, and possibly in the new kinds of things that Elena and I talk about, where you're actually physically improving your, it's not just about IQ, but you're physically improving, enlarging your cognitive icon so that you can exert compassion to much larger spheres of influence than you ever could before. Like that I think is a particular, it starts a feedback loop that I think takes you on a non-linear path. So there are things like that.
It's difficult to add something after Mike's explanation, but I will add just a small thing, that I believe that intelligence, it's not something sitting inside the organism, or in contrast, not something abstract sitting in a cloud. It's actually intelligence exists on the interface between organism, let's say, and environment. And the more complex environment is the more complex intelligence can be. So, and this is how basically learning works, right? We make our environment more and more complex, and we learn more and more, and we become more and more intelligent. And for that, you need to have this kind of proactivity, proactively reflect the environment and predict it.
Cool. I think we had two more questions. Angie and then Whit. Go for it.
Thank you, Michael, for your research. I was finding myself experiencing a lot of awe and curiosity and a lot of inspiration, but also slight trepidity around the subject. I understand that a lot of your research is around how do we guide specific outcomes with cellular intelligence. I often find myself asking more introspective questions around whether there is some sort of inherent intelligence within us that is driving the motivation to try to control or influence these cellular processes. As in, are there complementary research that helps us understand whether we are, whether that it's maybe an ethical framework, or whether there is a larger intelligence that's guiding our desire to drive these outcomes?
Wow. Yeah. I mean, so to really answer that is well beyond my pay grade. I think that's a very big question, and I don't know, but I do think that with the discovery of novel specific tools to uncover new kinds of intelligence within us and outside of us and all around us, in the ecosystems and all of that. And also, the more that happens, the more our minds are kind of open to the existence of these other things that are, I think, all around us right now and very hard to detect. I think those questions will start to, at least we'll have better questions. I think the answers will take a while, but I think improving the questions will start to happen fairly soon. Once we, one of the interesting things is that I think that you need a kind of impedance match between the tools that you're using and what you're looking for. So I think the reason physics doesn't see any of this stuff is because physics generally uses very low agency tools. You've got some rulers and some voltmeters and magnetometers and things like this. And low agency stuff sees matter, and it sees machinery, and it sees the hardware. What do we need in order to detect these other kinds of intelligence? Well, you need other minds, right? You need minds to detect minds, and so that's actually something that we're working on as well. Is to have living or engineered intelligence as the front end of our detectors or our sensors or our tools that we're developing to find these other intelligences. And I think once we start looking, we're going to find all sorts of just an incredible number of these things. In particular, certainly in the body. I mean, this is part of our regenerative medicine framework. Is this idea? I mean, this idea, like among alternative healthcare practitioners, this has been around for thousands of years. That hey, there's an intelligence in your body cells that is distinct from your own verbal intelligence. That seems at this point could just be completely unavoidable. That's true. I think that's absolutely true. And what are the capacities, the goals, and I don't think it's a single unitary intelligence any more than we are. I think there are a number of different beings to talk to in your body, and then to develop ways to understand. Okay, so how health and disease is one thing, but how do they relate to your psychological makeup, to your behavioral patterns throughout the day, to larger questions of psychoanalysis and spirituality and so on. I think all of this is going to be connected at one point. And one of my favorite quotes was from this is a paper that was in, I forget, it was at the Lancet or something. It was one of these major medical journals, and it was a woman who heard voices. She started to hear voices in her head, and the voices told her to go get a brain scan. And she got a brain scan, and sure enough, they found a tumor. And she had the tumor removed, and she was fine. And the last thing the voices said to her was, "We were pleased to," and I quote exactly, "We were pleased to have helped you. Goodbye." That was the last phrase she heard after the tumor was out. So now, right? So now you could. So what is that? You could think of it in a couple of different ways. You could try. I mean, one theory is that there's some other kind of intelligence that's involved here that's from somewhere else. But another, no less, and I think probably in a practical matter, even more interesting possibility is that what's happened there, which is rare but obviously not impossible, jumping of levels, and that what normally you, meaning the verbal, like our left hemispheres are having this discussion right now, so your verbal consciousness does not have access to your blood chemistry and all the weird things that are happening physiologically in your body. But is it possible, and we're actually trying to develop tools for this, to jump those levels and have a kind of conversation and push information from those levels up into consciousness, and maybe that's what happened to her, is that basically there was a physiological state. I was talking to somebody about this case, and they said, "Oh, well, that's just because she had the tumor was doing weird things with her blood chemistry and whatever, and she started hearing voices." Right, that's exactly it. That's the beauty of it, is that information from her body architecture got up into the verbal consciousness, and in a clear way that she was able to go get her scan or whatever. And so, yeah, communicating with these other intelligences that are there, I think, is a major opportunity for the future.
Excellent. Thank you very much, Michael. Just to put this slightly in perspective, because this might have been a presentation you wouldn't have expected, coming to this event. As Helen mentioned yesterday, a core part of our thesis about the artificiality is this idea that the synthetic and organic are blending. And the foundation of that is that information is common across. And you need information, computation, allows for intelligence and agency across all kinds of things that we don't normally think of. We've sort of grown with this idea, and I believe I'm quoting you, Michael, that we're a big bag of cells, right? And so somehow that bag of cells gives rise to these things that we think are unique to us. Well, how does that happen, right? And so Michael's work, Elena's work, is truly foundational to how we think about what it means to be human, what is the human experience of this new world, right? So thank you very much for all of this. It really sets the stage for our day. And we'll give a round of applause, but we're going to take a 15-minute break, so please get some coffee. But yes, thank you very much.
Thank you.